Can't get it out any higher. Anyway, use that one as well. That one. Oh. Okay. So, thanks for coming. My name is Roland. I'm going to hold this lecture and the next one, and also the lecture block at the end, because Lisa is not able to travel anymore. So you will hear me for the remainder of this unit. Apart from, of course, Mark Temple, he will give the section from from lecture 5 to 10 or something, unless he's doing it online. I can't remember now. But anyway, I'm doing that lecture block. I'm actually based in Parramatta. Okay, so if you have any issues or want to email me or anything about those lectures, you can see me there, or you can email me, which is the easiest. It turns out I'm also running my research lab with Lisa together. So we're both doing research on proteins, and I'll talk to you a little bit about this as well in that lecture. And we're also heavily using molecular biology in the lab, which I'll talk about as well. So first of all, the first 10 slides or so, I think I'll talk generally about genomes, theory and practice of cloning will be the second part of the lecture. If you do have any questions, please feel free to interrupt, raise your hand, ask me. I'm happy to answer any questions. Okay. So these are the learning outcomes for today's lecture. As you can see up there, you can read through those. It's basically to do with starting to understand how DNA can be assessed. What can I do with DNA? So the first things of cloning, that, as I said, this is going to be this lecture, next lecture, so we're talking mostly about cloning, so you get an idea what that actually means. Remember, this is one of the things you're going to do in the practicals as well, so it's a very, very important part of this unit. And you're going to do it, or you have already done it. You all have done the computer labs, right? Everyone's done the computer labs, so you have a good idea what it is that you're actually going to do in the wet labs. Only you did it in the computer. So you designed, if you want, you designed a vector. You had a piece of DNA that you took and then incorporated in a vector. We'll talk about this as well today. And then you were asked to determine how many of those fragments you get at the end. And you have to determine, obviously, what the PTS cleavage sites are, the enzyme cleavage sites are. For most of you, that was probably the first time that you've heard that, unless you have done genes, genomics, and human health. Who has done genes, genomics, and human health in here? One person. Who has done genetics? A few more. Okay, so maybe you have more. You have actually a good idea already what you're doing, which is good. But if you for some reason you're still struggling to understand the concept, don't worry. I'll go through it now in the lecture, but we'll also have a tutorial at the end. So we'll have 5. As you know, we have 4 wet labs, and then the last of those, so the 5th, is a tutorial where you can ask questions. So that's your chance. We're going through the whole thing again, explaining everything again, and you can ask questions as well, okay? And we have a little quiz as well at the end. So that's the tutorial. So you always have opportunities to ask questions, even in the lab, if you want to, but at the end, again, we'll go through everything again, and then hopefully it becomes clearer because you have done it in the lab. So you've actually worked on each step, doing each step. But the theory behind it I'll cover today and next time. Okay? Has anyone any questions that they want to ask me? Has anyone read the lab manual yet, by any chance? I'm very optimistic there. Yeah? Actually, most of you would have, because you had practice this week already, right? Yeah? Are there any questions that I can answer immediately that come to mind? So we'll talk about that. But first of all, let's talk about general, why do we need genes? Why do we need genome sequencing? How is DNA accessed? Why is this important? So obviously you can appreciate, and again, there will be, I think Lisa's already spent some time talking about this, sequencing, what methods you have to sequence. It is important to sequence genomes because this tells us a lot, of course, about us or about any organism that you sequence. But remember, it's one thing to have the sequence, which you obviously can use to determine differences between different species. You can build up so-called evolutionary trees, so you can determine how related we are to other organisms or how related so animals are related to other animals. That's all good. You can get the sequence. But don't forget, whenever I say that, don't forget that sequence is only one thing, and what you do with the genome is another thing. And that's very important because remember that, for example, our genome is only, I think, is 99% similar to the chimpanzees, but obviously we are humans and chimpanzees are chimpanzees. So it's not just the genes that decide how the organism at the end, how the phenotype looks like, of course, because there are things like gene transcription, okay, which you will talk about as well, or Mark will talk about it to some extent. So you have to use those sequences then to actually express those genes, to make those genes. So you transcribe first into RNA, and then you translate into protein. So it's very important which genes are transcribed, okay? So as you can imagine, 99% similar, but not all of the genes will be expressed, of course. So there's lots of genes that are not expressed, there's some genes that are expressed more than others. It's very, very different. And the way this is regulated, we'll also go through that in one of the lectures, Mark will. This is very, very important because this is obviously clearly a distinction between, for example, us and chimpanzees because they have different mechanisms of expressing these genes. So despite having the same sequence, not the same genes are expressed. Everyone knows what I mean if I say genes are expressed? Yeah. So it's, sometimes people also say genes get turned on and off. That's the same thing to say, okay, if genes get turned on, you're making protein. If genes get turned off, you're suppressing making protein, so you don't make protein. Another thing is, of course, not just the protein level, but also on the level of RNA. There's a lot of complex and lots of ways of regulation in there as well, so not even on the level of protein yet, so one step before that on the RNA level. So for example, we know now that, especially our brains, in our brains, the RNA levels are very, very important. So some of those RNAs are regulatory, some are not. So there's a lot of complex interplay between RNA and protein, which is very unique to our brains, for example. So this is also happens on a level that is not to do with the sequence, because the sequence is very similar, but it's happening on the level of RNA and then later on some of it at the level of proteins. So despite having these small differences, it's obviously important what you do with those genes and how those genes are regulated. As you can appreciate, because these DNA sequences are very long, small differences obviously contribute quite if you say a few percentage, that's a lot because we have long DNAs. Which reminds me, how do you think DNA is organized in cells? Is it just a linear arrangement? What do you think? No? It's packaged, that's right. It's packaged, that's right. It's packaged. Otherwise, it's not possible, guys. Okay, so if you think about DNA as one string, it would be so long, you could never ever fit that in a cell. Okay, so it's a couple of micrometers long, whatever, 10, 15. So you can never fit that DNA in there. It's impossible. If you work out what the length would be, I think the examples of wrapping around Earth or something for one DNA. So it's extremely long. You would never be able to fit that in the cell. So it has to be packaged up. We'll talk about that as well briefly. I will touch on that. But again, Mark Temple will talk a little bit more about on that as well. And actually, that leads me to the next one, which is understanding the difference between heterochromatin and euchromatin. So that's the two main states that your DNA can be in cells, or everyone's DNA, mammalian cells' DNA at least. And that means you have areas of DNA that is highly condensed, that we call heterochromatin. So does anyone, do you know how this, anyone have any idea how this actually works, how the DNA gets packaged up? What helps to package up the DNA? What helps there? Histones, yeah, that's right. So generally proteins, right? So there are proteins in there that kind of chaperone, or they help the DNA to fold up. And the chromatin, really, if you say chromatin, it's really everything. It's the DNA and those proteins, everything together is what we call the chromatin. And the euchromatin, on the other hand, these are regions of the genome that are loose, that are less packed, and that are open. So in this case, in the loose form, genes can be assessed, okay? So everyone knows, so I say genes all the time, but do you actually know what is a gene? What's a gene? Anyone can briefly describe what a gene is? Trying to engage a little bit, and you don't fall asleep. Yeah, that's a good description. That's excellent. Yeah, this is a certain area of gene that gets made into protein. That's right. Often it's used in many different ways. It's not quite clear, but that was a very good definition. Okay, people actually abuse it sometimes. They say things, genes where there's no genes. You know, if you hear slogans in the media like gene-free environment or something, it doesn't exist, right? Otherwise, if you live in a vacuum, there's nothing. But as soon as you have an organism, you have genes. Okay, so there's nothing like gene-free environment. Okay, so you can get something like if genes are manipulated in some way, then you can say the genes are not manipulated, but something like gene-free doesn't exist, okay? And there's lots of things like that in the media that you hear and don't always believe that. Yeah, so genes are turned on and off, and they can more easily turn on when you are in euchromatin, or in fact, you need to have it in this state that genes can turn on and off. So you can see already here, there's another level of regulation, which I haven't talked about before I talked about RNA and then protein. Actually, there's something in between the RNA and the DNA, which is called histone code, which we call histone code. It's another type of code that exists on the basis of histone proteins being bound to DNA, and those proteins regulate if the DNA is open or closed. Okay, only if the DNA is open, you can actually read the genes. So that's the first hurdle that the cell has to take is to actually unwrap that DNA to make the genes available to gene expression. That has nothing to do with then making RNA and then protein. That's the first step of regulation. Okay? And that, even at that level, we are very, very different, for example, to chimpanzees. Right? They have different regulation mechanisms than we have. So this is only this is not 100% understood yet. Of course, there's a lot of research going on at the moment as well. It's a very active area at the moment because, as I said, before we thought, okay, now we have the sequence. I don't know if you remember back in the 2000, whenever the human sequence came out, it was always, we have the sequence now, we can do everything. And then it turns out, actually not, because the sequence is not everything, right, as I mentioned before. It tells you something, but the way it's organized is from the sequence you can't see how it's organized. That you don't know. So you really have to dig a little bit deeper there and see how DNA is organized, and especially how this chromatin, how this looks like and how this is regulated in order to understand why, for example, we have certain genes turned on and other organisms don't have those genes turned on. And if you dig a little bit more inside that, so you can see how this DNA is organized in the cells. Again, I think Mark will spend some time talking about this. But the smallest unit that you actually find above the DNA is what we call a nucleosome. And you can see these little balls there. These are the nucleosomes. In this case, you've got 4. You've got the DNA in black, so the DNA is wrapped around those nucleosomes. And they always occur in 4s there. So and these nucleosomes are made up of those histone proteins. Okay, so there's certain type of histone proteins. I'm not going to go into detail now because it will be covered, or you might have heard about this, but there's histone 2, histone 3, histone 4, and so on. And those make up those nucleosomes. Okay, so they're like little balls on a string, if you want. And actually, sometimes people consider them as balls on a string or pearls on a string. And the interesting bit is they are actually moving around. Okay, they're not solid balls. They're not fixed. Because as you can imagine, if now you want to express the gene there, let's say the gene is just located here, it's very hard for any transcription factor or anything to come in here and actually turn this gene on. This has to roll out of the way if you want. And that's exactly what happens. There's a whole machinery behind it that activates that somehow, and then the balls, kind of see that here, they roll away, and then all of a sudden you have the DNA exposed. And you can express them, you can read the DNA and you can express it. Does that make sense? Yeah, so it's kind of the obstacles in the way, mechanical obstacles. And in fact, there are mechanical processes that happen there to push these balls out of the way. All right? And what is involved in that? Obviously, that's very simplistic here. One of the mechanisms that are involved is what we call histone acetylation or histone deacetylation processes. So you can see what happens there. So you've got these as I said, those balls are made up of histone proteins, and some of those histone, or actually all of them, have those tails. So they're called histone tails. So you can see these here. They're flexible, but that's a protein, okay? Remember, that's not the DNA, it's a protein molecule. So they have flexible tails. They have N tails. So the N terminus, remember, proteins have an N terminus and C terminus. So the N terminus is, in this case, it's the one that moves around. It's flexible. And those tails can be modified. So if I say modified, I mean chemically modified. And to understand that, obviously, now you need to know a little bit more about proteins, amino acid, which I hope most of you have heard already. So there are amino acids called, what are they called? What are the amino acids that are actually acetylated? Anyone's got an idea? No. Lysines, guys. Okay, lysines, amino acids that have an amino group at the end, they can be modified, they can be acetylated. So if you don't know what that means, please go back, consult your chemistry again. Are you first year chemistry or whatever you did it, or functional proteins and genes, done it? So it's those acetyl group that can be added on top of the lysine residues. And those acetyl groups here, you can see what they cause. They cause those nucleosomes to pretty much fall apart, to move apart and give way to those genes. So it's a very simple process. It's really just an enzyme. All it does, it adds that acetyl group onto the lysines, right? Lysines. Mostly lysines, can be arginines as well, but mostly lysines. And once that happens, it opens up. So it's really like a signal for it to open up. Okay, so it's one of those parts of that histone code that I call histone code is an acetyl group. Okay, the other codes like methylation, ubiquitinylation, somulation, I think, and there's a few other modifications that you can add to those tails, to those histone tails, that cause different things to happen. Okay, so that's what we call the histone code. If you've ever heard this before. And it's all regulated by enzymes. And sometimes, of course, it's not just the enzyme. It needs to have some other machinery to actually open this all up. Okay, it's not as simple as to say, okay, the balls move out. It needs more complex proteins and machineries that are involved that react to those signals. Okay, this acetylation is just a signal. It's nothing else. And because it's a signal that it's an acetyl group on your lysine, it's also something we call post-translational modification. Okay. Why do I call it post-translational? What do you think? What's post-translational mean? After the translation, that's right, exactly. So after the translation, not before. After the proteins have been made, in this case the histone proteins, they are modified. So they're post-translation modified, you add an acetyl group to them, and then you get that kind of scenario there. Okay, so that's what happens. But again, there will be more details in one of the next lectures. Okay, but that's basically one of the mechanisms that you have to understand in order to know why all these differences. It's not just sequence; it's more than that. Other things like that are different, of course, is rearrangements in whole chromosomes. Okay, so you see a chromosome here. You can over many years, obviously hundreds, millions of years evolution, things can happen. Okay, so things can happen. For example, you can have this would be, let's say, this is a gene that does something. Okay, that gene could be duplicated. So some process that we understand to some extent can make these genes duplicate. So that gene can arise on some other site in the same chromosome. Okay, so you have these processes. You have things like genes getting transposed to other parts of chromosomes. So you can have this part here, it's called a transposition. Or you can have things like inversion, so where one part flips back on the other one. And then if you have processes like inversion happening, you can imagine you have things like recombination. So you can have, now if you look at that, to understand it, just look at the white part, okay, and then you can kind of continue here, and you get this part. And if you look at from that side, you get that part and then the other part. Okay, so you get the other bit. So it's kind of taking parts from other bit and combining them with itself and then making two parts out of it. So all these things happen. And why do we know that these things happen? Of course, because one of the things is happening. And so have genes on one chromosome doesn't mean they're not getting expressed. You know what I mean? Like, it doesn't matter where the genes are in the chromosome, in the sequence, as long as they're there, as long as they're active in some way. Okay, another concept that I just want to briefly introduce is the concept of SNPs, which you will hear as well in one of the next lectures. SNPs are single nucleotide polymorphisms. So we have a single, that means a single base is changed. So if I say single base, what do I actually mean? If you think about it in the context of DNA, Is it just one base? Two, right? Because you got two bases. Remember you had double helix, so you got two strands, yeah? And a normal strand and a complementary strand. I hope that's everyone's got that. Remember when you did the computer lab, normally how we draw DNA is just from 5 to 3 prime, just one strand. We don't draw the other strand, but it's there. Right? Always think about that it's there. How about RNA? Does RNA have a second strand? No. Right. Okay. So RNA doesn't have that. Okay, so SNPs can be different, but normally SNPs are different. So, for example, if I look at my eyes and your eyes, actually I can't see your eye color, but I've got green eyes. I don't think you have green eyes. Blue, I think, yeah. So clearly your SNPs and your eye genes are different than mine. Okay, so eye color is one of the things that are different, which obviously doesn't really matter, really. So it's not a dangerous thing to have different eye colors. So normally SNPs are mutations if you want, but I wouldn't call them mutations because they're actually not dangerous in that sense. They can't cause a disease, okay? They're just variations that everyone has. Skin color, eye color, things like that are SNPs. So different SNPs, different people, different SNPs, and that can lead obviously to those things that I just said. And it's also that on the level of populations, they can carry different SNPs as well. So we can read actually what's I don't have to read this, but you have this thing called an allele. An allele is, so you have two alleles. So every time you have a SNP, you have two alleles. So you have there's a possibility of, for example, a C allele or a G allele, right? So in the C allele you have the C, in the G allele you have G. So there's always these two alleles. I'll show you an example here. So you can see these are 4 individuals here. The individual 1 has got the C allele here. So you can see here, you've got the option of between C and T. That's obviously something that people have to know, right? They have to sequence that. That comes from all the sequencing data that we have. But if you compare that particular gene of individual 1 and 2, or rather 1 and 3, you can see, for example, 1 and 3 differ there in that one base, which is a C or a T. So one is a C allele and a T allele. And if you look at it as a whole, in a whole population, you can say, okay, we've got 4 different types in a population. We've got haplotype 1 type, which got a C, a G, and a G allele. We've got haplotype 2, that has a C, A, and E, and so on. So we've got basically these 4 different possibilities. That's all that is. It's really 4 different possibilities of combinations of alleles, and you call them haplotype. Okay, so that's really what haplotype means. Okay, so why is this important? As I said, it contributes to variation. It contributes to things like different eye, different skin color, and things like that. What else do I want to say here? Yeah, so the other thing, the opposite thing of a SNP, if you want, is a mutation. It's not the opposite because it's really the same thing, but we call it a mutation if it can lead to some disease. Okay, a mutation has serious consequences. Okay, so if you get a mutation, if you have a mutation, it can lead. It doesn't have to, we'll see that as well. It can lead to diseases, okay? So that normally means a mutation is an area of a gene, which is important, of course, for something, right? In your development, or mostly it's development. So if you remember back in your computer lab, in the prac, are you going to introduce a SNP or a mutation? What would you say? A mutation, right? Why is that? Yeah, that disease that's right. It's this, can't remember, you can't even pronounce it, you can look it up, but it's the disease where you end up having these split toes and split fingers, okay? So it's a developmental disease. And remember, this is just one mutation. So introducing one mutation in that gene, and that leads to that disease. So why is that so serious? Why is that mutation so serious? Yeah, why is it not, you have SNPs that are not really dangerous, but why is that particular mutation that you're working on in the lab, why is that so dangerous? Do you know? Do you remember? It changes the amino acid, that's right, but yeah, so it changes an amino acid, that's right, that's one thing. So it's a mutation that changes the amino acid. We'll see as well that these are more serious than other ones, but let's say if it's an amino acid that doesn't play any role, then would it make a difference? If it's in a protein, okay, and it's in a region of the protein where it's at the end, for example, right? I would change it at the end, in the tail region. That is not really important for anything. Then it wouldn't matter, right? But that particular mutation, remember, that you're working on in the lab, is a mutation in the DNA binding site of that transcription factor, TP63. Remember, there's a transcription factor that you're looking at, and you're looking at the DNA binding site. So what do I mean by DNA binding site? It's the site that binds DNA, of course, right? A transcription factor, as you know, hopefully, has to bind DNA because it has to activate transcription, right? And to do that, it has to bind DNA, right? After all these processes that we talked before, that everything is in the open, the DNA is open, it can now, transcription factor can come in, bind, and it can't bind if that one residue is changed. If that one amino acid is changed from an arginine to a glutamine, it can't bind, okay? And if it can't bind, it can't do its function. So it can't actually do what it's supposed to do. And that's supposed to, I can't remember, it plays some role in developmental of some cells. And if you don't get that development, well, then you end up having these split toes and split fingers. So it's a very severe, very early on in the embryonic stage. If that transcript, like pretty much all transcription factors really have to work in the very beginning, right? When you develop an organism, of course. And if it doesn't work there, then you end up having these issues. And why do you think it's an arginine, by the way? Why is an arginine so important to bind DNA? Positively charged. Excellent, very good. Excellent. And why is DNA negatively charged? Yeah, phosphate backbone, exactly. So I'm asking these kind of things that, because they are important. And also when you go in the prac, you run DNA on gels, right? So you're running DNA. So remember, when you run DNA, it only runs on a gel because it's charged, right? You're applying a positive and negative pole, and it runs towards the positive or negative. Yeah, exactly. It runs towards the positive pole, that's right, because it's negatively charged. And it's negatively charged, like you rightly said, because it's a phosphate backbone. And also that's why the arginine, as being a positively charged residue, amino acid, it binds, we probably have to look at this, again, we have to look at the three-dimensional structure of the whole protein bound to DNA, that we can understand how it binds, but clearly the arginine is positively charged, so it's creating a big attraction, if you want, to the DNA. So if you change that by glutamine, then it's a neutral amino acid, so you eliminate that charge, and therefore it doesn't bind as well, okay? And that's why this is so serious. Okay, but now we're getting a little bit off topic, but we'll get back to this, get back to that as well when we talk about transcription factors. Okay, so we've talked a little bit already. You mentioned that it's changing an amino acid, and this is exactly what we would call a missense mutation. Okay, this is this case scenario, what you have in the prac as well, because that change in the base pair causes a change in amino acid. And why do I draw 3 here and not like 1 base pair? Yeah, that's right, because 3 bases is 1 codon, and a codon codes for 1 amino acid. That's exactly right. And again, we'll talk about, Mark, I think, we'll talk about translation as well, so we'll talk about genetic code and so on. So we'll touch on that, but just keep in mind you need 3 bases. Why do you need 3, for example, not 4 or not 2? What do you think? Without knowing anything about codons, if you have 2 bases determining the type of amino acid, how many possibilities do you have? We have 2 bases. One base can have you 4 possibilities. The other base has also got 4. So 4 times 4, 16, right? So you've got 16. If you had a code that had only 2 bases, you had 16 different possibilities. And how many amino acids do we have? Yeah, 20 something. That's right. So more than 16, of course, right? So it's not enough. So to have 2, you just simply don't have enough possibilities. If you have 3, you have how many possibilities? Oh, it's going maths now. 4 times 4 times 4? 64, right? So that covers everything. That's enough. You don't need more than 64, right? So even if you had 4, you had 64 times 4, whatever that is. So that's too many. So it turns out 3 is exactly the right number to cover all the amino acids, okay? So that's what that is. So if I change one of those bases, you can see this colon GCA now changes to TCA, and it turns out that GCA codes for alanine and TCA codes for serine. So what you're having here is an alanine to serine. So it's a change of the type of amino acid. So that's very serious, okay? Yeah. Having said that, of course, it depends where that mutation is, of course, right? As I said already, if the mutation is somewhere in a part of the protein where you don't really need it, then it doesn't matter. But if it's in a region where the protein, where it's important, like in your case, in the DNA binding part, it plays a big role. Okay, so that's why it's important. On the other hand, you can have things like a silent mutation. Okay, so you have, in this case, CTA to TA. So what you're doing here is you're not changing the type of amino acid. You're not changing the type. So the amino acid stays the same. So that's a lucky one. Okay, if you have one of those, you're lucky because it doesn't do anything. Okay. Whereas if you get one of those, that's very serious. Okay, because from the TSC to the TAA, what it does, it creates a stop codon. What's a stop codon? Yeah, it stops. So actually, that's right. So that's the name suggests, it stops. So transcription stops here. So or translation, so transcription, and then obviously you end up having a smaller protein. So if you have normally a protein that consists of 1, 2, 3, 4, you then have a protein consists of 1, 2, and a part of 3, and that's it. So you actually have a truncated protein. So you're losing a part of the protein. That's a bad thing, right? Because if you then fold the protein up and you have a three-dimensional structure, the protein might not fold up anymore because it misses a whole part. And if it doesn't fold up, it's not functional, okay? Therefore, cell is dying. That's it, okay? And in the worst-case scenario, your cell doesn't die, cell replicates randomly, and you get cancer. Okay, so those type of mutations can lead to cancer. Same for these ones, if you get a lot of them. But remember, it's not that bad because you have a DNA repair machinery, which we talk about at the end of this, pretty much at the end of the lecture series. Okay, so you've got a whole machinery in your cells that there's a mutation, go in there and fix it. Okay, so it fixes it. So it's not that bad. The problem is if it's overwhelmed, if you have so many mutations all at once, then it's a problem because then the DNA machinery can't catch up anymore. Worst case scenario, you get cancer. Okay? So for example, you go out in the sun, not today, today is harmless, but in a 3 or 4 months time when the UV index goes up to 9 or 10, you go out, you stand out there in the sun without sunscreen, the sun, the UV is damaging your skin in the sense that it damages the DNA of your skin. Okay? So it goes into your cells of DNA and mutates, we'll see that as well when we talk about this, it mutates your individual bases, and you have mutations in there. Okay? That's a bad thing because then those skin cells don't work anymore. And if you can't fix them in time, you get what's called a melanoma, and that can then spread, and you know the rest. So that's not what we want. So these are dangerous mutations that we can have. Okay, and going back to the SNPs, that's probably the last step before we talk about the cloning thing. You will probably realize I spend a little bit of time on this, which is good for you. Because if you do the summer, I normally do the summer series, in fact, and the summer lectures are 3 hours, and I spend 3 hours to do 2 lectures, okay? So then I don't have much time. So consider yourself lucky because I can spend a lot of time. And then you can ask, or I can ask you. In summer, this is a bit harder. Anyway, so you can see that what happens, so just because you have a certain SNP in this case it's actually, I would call it a mutation rather than a SNP because it actually leads to a disease, or it can lead to disease. So if you have that particular allele, which is a high-risk allele, you have a probability of getting this disease of 78%, pretty high. But on the other side, if you don't have that allele or that mutation, let's call it mutation, you actually still have a frequency of 36%. So, okay, it's about double that, right? You have a double the chance. But, okay, that really doesn't help you. You still have overall a third of chance that you get it, right? So it's always interesting to look at the absolute numbers and not just of the percentage, because it's always misleading. So you still have 36% chance to get that, okay? That just tells us that it's either another gene that plays a role, another sequence somewhere that we haven't found yet, or B, environment, okay? And of course, that always plays a role, okay? So just by having a mutation or in the right place doesn't mean you get the disease. There's a high chance, but remember you've got environmental issues that often play more roles. So for example, obviously, lung cancer is a typical example. You can get lung cancer because you have a genetic mutation, but that's very rare, right? It's more often that you get lung cancer because you smoke, okay? So it's the environment that plays a much bigger role. Okay, does that make sense? So even though you know the sequence, it tells you something, but it doesn't tell you everything. So if someone comes to you and says, send me your DNA, I sequence it for you, and you can do that now, you can send that off, it's pretty cheap. You can do that, and it's informative, and you get some information out of that, but it's not everything, okay? As I said at the beginning, the sequence is the sequence, and what happens with it is a different story, okay? So don't think because you have a certain mutation is that, Obviously, it's useful to know. So this information we didn't know before, right? And one of the examples that I always talk about is, and you probably heard of this, is the BRCA gene in women, breast gene. So if you have that particular mutation in the gene, you have a very high chance of getting breast cancer. And knowing this in advance is a good thing because you can do something about it, right? I mean, you monitor anyway, but you can monitor it more, or in worst case scenario, remove your breast, then you eliminate the risk altogether, right? And people do that. But that's obviously you can only do that when you know what the sequence is, if you have that mutation, okay? So that's an important factor. Okay, that slide you have seen in the prac notes. So you know, we've talked about this. This is what you're going to do in the lab. So you're going to introduce that mutation there. Obviously in the computer lab it was easy because all you did is you took that sequence and you changed that one base. How are you going to do it in the lab? Use primer, excellent. Use primer. So the primer has got the mutation in there, that's right. So you're introducing a mutation by introducing a primer that binds. What's a primer? Can someone from over there maybe have a go? I always look at you guys. Anyone knows what a primer is? No, primers? What's a primer? Is a primer DNA or RNA? Who is for DNA? Hands up. Who is for RNA? Oh, that's funny. So everyone in here is not for DNA, not for RNA. So what is it then? So it's a DNA, it's a piece of DNA that binds. And why does it bind? Sorry, I have to go back to you because you're responding. Yeah, it's complementary, that's right. It binds because it's complementary. And you know, remember you would have looked in the computer lab and you looked for your primer sequence. You looked at where the primer sequence anneals in your DNA. So that's exactly what the primer does. It anneals. Annealing meaning forming these hydrogen bonds between which bases? What's the complement of G? What's the complement of T? A, that's right. So AT and GC, so these are the complementary bases. So you need to know these guys because you will encounter that in the lab. You will read your lab manual and you'll go, why do I use this sequence? What's the sequence? Or maybe you won't because you don't care, but if you actually care, you look at the sequence and say, where is this coming from, okay? Where is this sequence coming from? It's exactly the complementary sequence of my gene of interest, okay? And the other primer that you're using is the reverse complement. Remember, it binds on the other strand, but it's also complementary. It's just reverse complementary. Okay, so it binds there and anneals. And that's how we introduce that mutation. Okay, we do that in the lab, and we're doing a few steps. Primer, we use primers. We're using a thing called PCR. What's PCR? Yes, excellent. So remember, you have to submit an assignment on that as well. So now is the time that you look at what PCR means, okay? Polymerase chain reaction. What do I need for PCR? How does PCR work? Because you're going to do one in the lab. And if you already know how it works, it's great because you can say, oh, you put this and this. And what also is good, if you know how it works, you can't mix up primers. And that's the thing that always happens in the lab, okay? People always mix up primers and then they don't get a product. And then you go, oh, what happened? I don't have a product. So yeah, which primers did you put in? Yeah, this is it. Oh, the wrong primer pair, okay? So you need to understand which primers you put, how PCR works, and then you can understand what you do there. But hopefully you will when you do it, or at least afterwards. And that pretty much leads me to the cloning part, okay? So cloning, one of the main things is when doing cloning is we do PCRs, because PCRs are easy. I can design primers, I can use primers. You've seen have you done a PCR yet? No? Yeah. You did one, okay, good. So you know the little machine that you put it in, it heats up and so on. So it's pretty easy, pretty straightforward. That's why we use it a lot when you do cloning. And what can we actually clone? Okay, so we can clone genome DNA or cDNA. What's the difference? Yep. Yep, that's right. And genome DNA is everything, right? The whole chunk. Although people used to call it chunk, it turns out some of it is actually not chunk. Okay, there's lots of regulatory sequences that are not translated, but that doesn't mean they're not important. Okay, so that's also one thing that you really have to get in your heads is just because it's not DNA that gets translated in protein doesn't mean it's DNA that is not important, right? Because that chunk DNA often is regulatory and often plays a big role in how the whole organism works. And why do we want to clone these types? Well, there's lots of reasons for that question, right? And obviously in your cases, you want to introduce that mutation. But why do you think, why would you want to clone DNA? Can you tell me a reason? I mean, there's lots of reasons. Do you think, can you tell me one reason maybe? Yeah, study diseases, yeah, that's right, yeah. So if you know, for example, you know from studying a certain phenotype of cells, you know the disease has this effect, right? So you then study, you find it's a particular protein that causes the disease, then you go back into the sequence and you find out, okay, how does this cause the sequence? So then I can go back to the sequence and take the sequence and clone it into something and then study it in separation, right? Rather than the whole cell, I can take it out and study it in separation. So that's one reason. Another reason? Yeah, it's another reason, that's right, transgenic organisms. Or detection, right? All the forensics people amongst you probably know this. If you have someone else's DNA, you want to find out who that person is, right? You have to clone it. You have to take the DNA, clone it, and then sequence it. And because sequencing you can only do once you clone it and you amplify it, because you need a lot of it, right? If you want to sequence it, you need a lot of it. And that's one reason to do that, is to clone it and then amplify it, to make a lot of it. And we see that as well. You do that in the prac, you're amplifying DNA, okay? So there's basically two ways of doing this in the lab. One way is the PCR, and one way, what's another way of amplifying DNA? Oh, that's the sequencing type, to amplify, to make a lot. So you can PCR it, or in your case, remember, one of the steps that you're going to do is you're taking, once you've cloned your vector, and it's finished, you transform it into an E. coli bacteria, and the bacteria grows, right? So you're making a lot of E. coli, and because it's making a lot of E. coli, it makes a lot of DNA. So really what you're doing there is you're amplifying the DNA, okay? Why do you grow bugs to amplify DNA? Why don't you not just do a PCR? Yeah, that's a step before that. So you're selecting for the ones that have the vector, that's right. But could you not just use, like you introduce your mutation, you have your DNA, and now you just want more of it. So now at the end, I could just have a start primer, an end primer, and I could amplify everything. But the reason I'm not doing this is PCRs are error prone, okay? So you get errors in there. You can have errors and create errors, and that's exactly why you have to select for them as well, to afterwards make sure you have the right clone. But if you transform it into E. coli and you grow the E. coli, there's a much less chance of getting errors in there. So that's really why you grow them in E. coli and not PCR them. Okay, CDI is what you rightly said, is the DNA that gets actually transcribed and translated. So if you think about it in introns and exons, remember these are, the exons are getting the ones that are getting made, the introns are the cut out. So you've got these 1, 2, 3, 4, in this case 5, 6. So it's really those ones that then gets made into protein, okay? Not the rest that's in there. And how do you get to that? Well, here it is. You're doing a thing called reverse transcription process, okay? So what's reverse transcription? Obviously, reverse transcription is RNA, protein. Well, actually, sorry, transcription is only DNA, RNA, right? So reverse transcription is going from RNA to DNA. Okay, so in this case, we actually have RNA, and that's exactly what who's done the prac already? So the first thing that you did on first day, number 1, is a reverse transcription reaction, right? So you got given RNA. Remember, that's the thing you got, the tube with RNA, and then you make DNA out of that. And why do you do that? Well, we do that because the RNA is actually the one that you're interested in, because the RNA is then that sequence that gets then, it would eventually get translated. So that's our cDNA, our template for making cDNA, okay? So then you're taking this RNA, you're reverse transcribing it back into DNA, and you have, in that sense, you only have the DNA that was originally made to RNA, if that makes sense. Because mRNA you only get from those genes that get transcribed and then translated. Does that make sense? Everything else you don't get mRNA from. So really what you're getting through that is a cDNA, what we call a cDNA library. Okay, so it's really only that part that gets then made into, would have been made into protein. And so how do you do that? Well, you know that mRNA has got these poly A tails there. So you actually have to have a primer in there. And there you go, that's a primer, T. And Niels obviously nicely because you've got A's and T's there. And then you can make your, first instance, you can make your DNA, and you then degrade your RNA, and then eventually you make, to get your double strand, you have another primer there, and you're making a double strand RNA. You duplex DNA. So that's then cDNA. That's your cDNA. So what's the business about the length here? Why do we need a certain length for the primer? I hope people are not leaving because I'm asking you too many questions. Yeah, that's a good point. So if it's too short, it would anneal somewhere else, you mean? Like it's not specific, that's right. It needs to have a certain length to be specific. But more importantly, it needs a certain length to actually stay on there, okay? Because if it's too short, it will fall off. Why is it falling off? Because if it only forms, 4 or 5 base, for example, there's, you can work it out. There's 2 hydrogen bonds per AT and 3 hydrogen bonds per GC. So if you only have a certain number of hydrogen bonds, the whole thing is not going to stick properly, right? So it's going to fall off. So if you heat it up in your PCR tube, it's falling off. So it needs to have a certain length to stick on there. But then it can't be too long, because if it's too long, it sticks too good, right? And then it doesn't come off anymore, okay? So you need to have this balance between it coming off and annealing, okay? So what we also call the melting temperature has to be set in a way that is ideal in the PCR reaction, okay? So if I what do you think? If I put in a G in my primer versus a T, what would make a bigger difference? A G, because it adds 3 hydrogen bonds, right? Makes sense. So a G would increase my melting temperature by more than a T, right? Okay, excellent. Okay, so that's kind of what you need to understand if you talk about primers. And I'm sure if you're researching PCRs, you will come across that, how to make primers and what's important and so on. But that's, yeah. Okay, so once we've got our cDNA, we can then use that cDNA. We can call it a cDNA library. So a cDNA library is a collection of DNA clones, for example, from a genome or from an organism that I can use. In your case, by the way, there were skin cells, okay? I think you had skin cells. So we actually had skin cells in the lab, and we then, or maybe they weren't skin cells. I can't remember. I think they were skin cells. Anyway, there was a certain type of cells that we made the RNA from, okay? And then we froze the RNA, and you got that RNA. So you have a mix of pretty much all the genes from that cell, right? Remember, in that tube that you got in the beginning, you have a mix of everything. So it's not the gene that you want. You actually have to get the gene. And the way you're getting it, again, is you're having a primer, a specific primer that anneals to it. Remember, so you're kind of fishing for it, okay? So you're fishing for the gene of interest that you're interested in. What is that gene? TP63, right? So you're looking for that TP63 gene. So you've got primers that are specific to the TP63 that you looked at in the computer lab as well. They're annealed there. And so all you're making in your case is a DNA of your TP63. So you're creating a DNA clone of your TP63. All the rest doesn't matter, right? You get rid of it. It's really only the TP63. So does it make sense? Are you looking a little bit confused, no?
I was just thinking about how difficult that would be before you knew what the sequence you were looking for.
Ah, yeah, that's a good question. Yeah, if you don't know anything, it's impossible, right? So you need to know something about that. It's exactly right. And that's why we're using these sequences like these A's, because we know they're always there, okay? So we can use those. But if you don't, you can use those, but if you actually want to get a specific gene you out, you need to know something about that gene, the beginning or the end. And so if you do forensics, if you're looking like, if you're taking a hair, right, and you sequence a hair, so you know what a human hair, what the sequence is, right? So you have a lot of information already. So if you just want to see if it's my hair or your hair, right, if we both of us suspected of a crime, let's say, right, and they would know one of us does it, well, clearly we have a lot of genes that are similar, right? So you can use those similar sequences as primers and you're only PCIing up the parts that are different, right? Which is kind of the fingerprint, if you want. You know what I mean? Like that would be your hair and my hair, that's the difference. But there are areas that are the same, and that's what you would use. But of course, you have to know this, that's right. Okay, and so this is just an overview of the whole process, how cloning works. What's the time? 1. Do you want to have a little break, short break? Hopefully no one else runs away and everyone gets... So maybe let's do 5 minutes or 10 minutes. Maybe let's do 5 minutes. 5 minutes break is fine, yeah? Okay, and then we move on from there. Okay. So this is the overview. That's how we normally do cloning. Having said that, it's just a simplification, of course, as usual, but it describes fairly nicely what the individual steps are. But there's one important difference, of course. So if you have already looked at this, you probably might have noticed this. In your case, so when you work in the lab, you're not going to cut out the whole chromosome, of course. So what you're doing there is actually you're amplifying a certain region, right? As I said, so you have an mRNA. In this case, this would be mRNA, and you're having primers specifically to that region in red there, and you're making only the red part and not the blue part. Okay, so you're making a specific part, that TP63 gene, the DNA binding part of the TP63 gene, and then you're ligating that in a vector. So this is the same from here onwards, but this would be different. Okay, but it's normally it's a standard thing to have some sort of cloning vector, which is a plasmid. Why is it a plasmid? Why do you think it's a plasmid and not a linear? Size of it is easier, yeah, and it turns out that E. coli, if you give it a linear DNA piece, it will just degrade it, right? It's not going to go in there, and if it goes in there, it will just be degraded. So it's not going to be incorporated into the host organism, right? Which is what you want if you want to replicate it and amplify it. So it is circular in all cases, and it has got. We will look at that in a minute what it can look like, but simplified, you cut that. And you insert your gene of interest, and then you transform that into a host cell. Okay, so can it be anything, but in our case it's E. coli. So we're using E. coli. Remember, E. coli is a very simple organism. It's not very dangerous. It's where do you have E. coli? In the guts, yeah, in the intestine. That's right. So not having said that, there you can't. I mean, you wouldn't swallow them, right, and drink them or whatever. That's not a good thing. You can get infections, of course, or spurt them in your eyes or whatever. That's why you wear gloves in the lab coat. But they're fairly harmless in the sense. They're not, they're fairly harmless. And the other advantage is, of course, that we know them very well. They've been studied to death. Okay, E. coli, they know everything about E. coli. They know the sequence. We know everything. So that, of course, is useful because we can take that and manipulate it in the way we want it. Okay, so in this case, it's a certain strain and has been modified, the strain that you're using, in the sense that it's easier for those strains to take on the DNA and they replicate quicker, and I can't remember what else, but there's a few other things that they have changed and modified. So it's ideal for your purposes, for cloning purposes. Okay, so it's not just any strain not the strain that you have in your guts. It's a modified strain. Okay. And so those they take that on, obviously, to some extent, and then they grow. And then you let them grow. In your case, remember, you have plates, and they grow on plates. Okay, and so this is what a vector can look like. So this is an example of one vector. So just briefly go through these different areas here. Origin of replication is something we need, obviously, to replicate. So that's an important one. What are all these things here? What does that mean? BST1, EcoR1, BAMH1. What do you think that these, what does that mean? Yeah, exactly. They're restriction sites. So that means what? Yeah, perfect. So that's the sites where those enzymes can bind, right? So there are certain enzymes that can cleave, or when I say bind, it first has to bind, of course, and then recognize that site. It means it has to specifically bind. Okay, so the enzyme has a binding pocket that specifically recognizes that sequence here. Okay, you don't see the sequence. All you see is that name of that enzyme. But I can assure you that there's a sequence here that is the recognition sequence of that enzyme called PST1. Okay, so it comes in, binds, and then it cleaves. So it cuts the DNA, cuts the phosphate backbone. Okay, so it cuts the DNA, and that's what it does. And then you have a recognition sequence side of EcoR1, BAMH1, and so on. So why are these useful? Because it allows you to specifically cleave at that position. Okay, so if you have a gene that you want to insert, for example, here, right? So then you would have to cleave with BAMH1 and SAL, I think that is, SL1. So then it cuts out this piece, okay, of DNA, and you can insert your gene of interest. But of course, for gene of interest, and we get to that as well in the next lecture, but remember, it doesn't just go in like magically, right? It has to go in with the same recognition sequence that enzyme cleaves out in the first place. Okay, so you've got sticky ends, and the sticky ends have to be complementary, and then it goes inside. Okay, we'll look at that later, just to get the idea. In your case, in your prac, are you using those enzymes or not? How do you get your TP63 DNA into your cloning vector in the prac? Sorry, I can't hear you. BAMH1? No, in your case, you don't use BAMH1. What do you use in your case? The PDS1 side you don't use to get it in, but you're using it afterwards to check whether it's actually in there, that's right. Now, in your case, you don't use any enzyme. In your case, you use that what we call the TA cloning. Okay, so TA cloning is simply an overlap, an overhang of T's and another overhang of A's, and so then the piece moves around in solution and then finds itself right in complementary basis. So it just goes in, and you don't need an enzyme. Okay, so you're getting these overhangs. In fact, how you get them in is when you PCR your gene, the PCR is done with a polymerase. That's the enzyme that adds the bases. And that polymerase, when it's finished, at the end it adds an TT overhang. Okay? And your vector itself has an A overhang, and so that's how it then finds itself. Does that make sense? Remember when we talked about the computer lab, it can go in this way, but it also can go in the other way because of that TA business. If it was a restriction enzyme, it would be clear cut, okay? It would be BAM, echo. Then it would go in this direction. That's what's called a unidirectional cloning. But if you have a TA cloning, like in your case, it can really go in either way, right? So you don't know that. So you will have probably 50-50, or we'll see when you do it. But that's the reason why it can go different ways, because you have a different way of cloning it in. So you don't need restriction enzymes, but disadvantage, of course, is it doesn't know which direction it goes in. Okay? And that can be important, of course, later on. If you just use the DNA, that's fine. But if you actually want to make protein from that, you can imagine that if you put it in this way, or putting it the other way, is making a huge difference to which protein you're going to make at the end, right? Does that make sense? Because the N terminus, C terminus will be swapped around, and whatever you're making doesn't make any sense, right? If it's going in the wrong way. Whereas in this case, if you make DNA, it doesn't matter because you just make the DNA. Okay, well, again, we'll talk about this a little bit more in the next lecture, just to give you a hands up to that. So this is what yours look like. So here's a real world one if you want. It's called PCR 2.1. That is commercially available, so we buy that from Sigma, I think, or I can't remember from where. We buy it, and then you get it from us, okay? And as I said in the beginning, you see here, you can see here these TT and these AA here. There's two A's, two T's. That's exactly what happens. So your PCR product, your amplicon, your gene of interest, your TP63 gene is the black bar, and then you have the vector around it, and the vector itself comes already open, okay? So if you get your vector in the lab, it will be already open, okay? So you don't have to open it up, it's already open for you, and all you have to do is take that vector, put in your PCR product, and it will then insert itself, of course. Of course, you need a ligase as well, right? You need an enzyme that helps you making the, because remember you have to make a covalent bond because it has to, the phosphate backbone has to be closed, and that enzyme that does that is called a ligase because it ligates that. It connects those end bits that you want to connect. Okay. Okay, and the other things as well in there. Let's talk about this one, ampicillin and kanamycin. What are these guys for? You have any idea? Antibiotics. That's right, the antibiotics. So anyone that's ever been to a actually, I don't know if there's sometimes have other names, but sometimes if you actually look what you're taking or when you get antibiotics, I think ampicillin is still given, but I can't remember, but at least the derivatives of ampicillin these days. So this is an antibiotic. So why would you need an antibiotic? What does that actually mean there if it says there? So it means that this part of DNA here, it doesn't mean it codes for ampicillin, right? That would make sense. But what it codes for, so if I say codes for, that's a sequence of a protein, okay, of an enzyme, in fact, that is able to cut up ampicillin. Okay, so that's an enzyme that can cut up ampicillin. So that means that part in the vector, if it's going to be expressed, it's going to be made in a protein, it's creating an enzyme that can specifically cut up ampicillin. So it's an ampicillin resistance gene, what we call ampicillin resistance gene. So why would I need that? Yeah, exactly. That's exactly right. That's exactly right. Very good. So it's a selection process. So I want to select for E. coli, for bugs that have that plasmid, as opposed to ones that don't have plasmid. So if you look at this process here, that's ideal, of course, because you can see the plasmid goes in every single host organism. But of course, you will have bugs that don't take on the plasmid, right? Because it doesn't go in there. If you do the transformation, it's not perfect. Okay, some bugs are getting the plasmid, some are don't. But the ones that don't have it, you don't actually want. So you only want to amplify the ones you have it. So an easy way of selecting for those bugs that have it is include something that is a resistance gene, which really means it makes something that can then selectively chew up or cleave up ampicillin. So if you then grow your bugs in ampicillin, all the bugs that don't have the resistant gene, so all the bugs that don't have the plasmid are going to die, of course, because antibiotics. And the bugs that have it will survive because they will pretty much be able to combat that enzyme or that ampicillin. Okay, so you will find when you make your plates, and also when you grow at the liquid culture, but in your plates, when you make your plates, there will be ampicillin in there. I think it's ampicillin in your case. I forgot, but it has to be one of the antibiotics. So you incorporate that in your plate, in your matrix when you make your medium, your LB medium. Remember the LB medium that you have to make. What's the LB for? To grow them, right? So nutrients for the E. coli. So that's it. But there's also antibiotic in there, okay? And there's other things like Excal and IPTG in there, okay? So those three things you have to put in. So now when you get the prac manual and you read it and you do that, then you, of course, because you've been to the lecture, you know why you need these three things, right? Actually, you know why you need one thing, because I haven't talked about the other two things yet. But one thing is ampicillin. That's exactly because you want to select for those colonies, for those bugs that have the vector in there, okay? And you can use, in this case, you've got two of them, you've got kanamycin and ampicillin, so you can use two, it's up to you really, or you can use both, right? Then you have an even more stringent selection, what we call stringent selection, so you have two antibiotics that work better than one, okay? So then you really make sure you kill off everything that doesn't nearly have these two things in there, right? But in your case, in the prac, I think you only use one. Okay, so that's for the selection. Then you have another thing called the LEGZ gene. What's that? What do you need that for? You probably haven't heard of this, but this is what's called the LEGZ overrun of E. coli. Again, this is something typical to E. coli. People have studied, and we'll talk about it when we talk about gene regulation, or Mark's talking about it. But in this context, it's interesting and it's important because you actually use this gene here, this LEGZ gene, you use it for selection as well. So using that for selection, but you're using that as well for selection. So that LEGZ gene, you're using for this type of selection. Okay, you can see here what selection I'm talking about. I'm not talking about a selection based on the fact that you have the plasmid. Okay, all these, in all of those three instances, you have E. coli that have incorporated the plasmid. Okay, so you already selected, you already killed off the ones that don't have the plasmid. However, you can still have these three different cases, right? You have a case where you insert your DNA exactly where you want it, or it's somewhere outside of that region, or it's not in there at all, okay? But in all three cases, you have a plasmid. So in all three cases, remember here you have the ampicillin resistance. So in all three cases, you would get growth. So you get colonies, okay? So that's one selection. But we need another selection to make sure that our DNA of interest has actually inserted itself into the right spot on the plasmid. And the right spot is here. The right spot is exactly in between the LEGZ. Okay, so here's the LEGZ gene. It turns out it inserts itself right in the middle. Okay, and this is important to understand later on because it actually disturbs the gene if you want. So you can see it's right there, and that LEGZ gene is disturbed or disrupted. So okay, so you insert another sequence in an already existing sequence, and of course that creates havoc with the original gene, right? The gene is not the gene what it was, and it won't do it won't do what it's supposed to do, and that's creating the LEGZ, the product of the LEGZ, which we'll talk in a minute. Okay, so it's basically in there, whereas here it's outside, and here it's not in all. So in these two cases, the LEGZ gene is still functional, okay, and what you get at the end is blue color. Okay, so you're getting colonies that have blue, color, and in this case as well. And in this case, because there's something in that LAC-Z gene, you're getting white color. Okay, so when you look at your plates in your prac, when you do them, when you grow your things, then there will be a week or two in between, and then you get the plate back where the colonies are on there, and you look at the colonies, and you will hopefully see you have a subset of blues and a subset of whites. It's normally always a mix, okay? It's never one or the other, otherwise something's wrong. But normally what happens in reality is some people have no colonies, and some people have only blue ones, and then there's some people that have a mix. Okay, so you really want the mix, because then you can pick the white ones. That's the ones you want, because that's the ones you know have the PCR product or your gene of interest in the right section of the plasmid. Okay. So it's another selection. And how does the selection work? Well, it's a little bit hard to understand. And again, I go through it briefly, but we will talk about it again, or Mark will talk about it again when we talk about the oberons. But the way it works is you've got this is the set of, that's the DNA there. There's a LAC-Z gene, okay, so that's LAC-Z. And there's the LAC-L. So this whole thing is called the lactose oberon. Okay, so lactose oberon is an important oberon. It's an important way of how genes are regulated in E. coli, and we can borrow this kind of. Okay, so we can often people take this part from the E. coli and borrow it for some other application, for some other cloning application. In fact, when we clone our proteins in our lab, in research lab, we have this oberon as well in our vectors. So we borrow that, but it comes originally from E. coli. So that's what I said in the beginning. E. coli have been studied so well, they know lots of things, everyone knows a lot of things about them, so we can actually borrow things from them that are useful for us, for our purpose. And in this case, we borrow it in the sense that we're having it present in our vector, and if it gets disturbed, as you can see, if it's disturbed, it can't express that gene, that LAC-Z gene, right? But to go a step back, that LAC-Z gene is actually not being expressed in the first place, because what happens normally is there's a sequence before that makes a protein called the LAC repressor, which binds to that promoter here. So what's a promoter, by the way? Do you know what a promoter is? Yeah, that's right. That's right. Yeah. So those type of questions can be an exam, by the way. So I can ask that, and you get an idea what we ask you. We don't ask you normally. We don't ask you. Yeah, we ask you things. If you understand the whole concept, then you can answer them. That's kind of one of the questions. Okay, it's not really, because it's just the definition of a promoter. But to understand how it works, you have to know, okay, it makes this repressor, which obviously represses genes. So it binds to the promoter, and by binding to the promoter, it blocks the gene. So it blocks that LAC-Z gene, okay? So really, that LAC-Z gene is blocked. So you wouldn't express it anyway. So then you might ask, okay, why do I then disturb it if I don't express it, right? The answer is simple. You actually get rid of that LAC repressor, because you, in your prac, on one of the three things you add to your plate, so we've got ampicillin, okay? You add IPTG, and you add XGAL. So IPTG is one of those molecules you add in order for that LAC repressor to be blocked and not bind to the promoter. I know this is a 2 negative now, but that's how it works. So the IPTG binds to the LAC repressor, and then the LAC repressor can't bind to the promoter, so you have expression of the LAC-Z gene. Okay, so you would get the LAC-Z gene expression if it wasn't disturbed, of course. If it's disturbed, if your clone of interest is in there, it can't express. But let's say you are in one of these cases here, where you have it either there or not at all, then your LAC-Z gene is undisturbed, and it can then be expressed, but it can only be expressed if you block this repressor, that in turn blocks the promoter, and you're blocking this repressor by having IPTG in your plates. Okay, so IPTG binds to that LAC repressor, and then it can't bind there, and you have the LAC-Z gene expression. And what that LAC-Z actually stands for, what it expresses, is an enzyme called beta-galactosidase. Okay, so that beta-galactosidase is the enzyme that you're making. And so in your case, as I said, IPTG blocks the repressor, so you would make that enzyme. And if you have that enzyme in the presence of XGAL, this is this molecule here, which is the third thing that you add to your plates. So now you really know why you have to add these three things, right? Because you have XGAL, ampicillin, and IPTG. So IPTG induces the LAC oberon, so it induces the LAC-Z gene. So you would make that enzyme there, that beta-galactosidase, and the beta-galactosidase, it works on that substrate. It's an enzyme, it works on that substrate, and that substrate, when it reacts with that beta-galactosidase, okay, it gives you that blue color. Okay, so you get a blue color if you have this reaction. Okay, but remember, if your clone is in the right spot, which is the LAX-Z gene, then the LAX-Z gene can't express anything. Even though it's turned on by IPTG, it's not expressing anything. Okay, because it's not making this enzyme, that beta-galactosidase. And if it doesn't make that enzyme, well, if the enzyme is not being made, that XGAL is not going to get a blue color. It's going to stay white. Okay? And that's a lot of stuff, I know. Go for it again, let it sink, and then ask me again, or we'll go for it again anyway. But that's basically the way of how we select for colonies that have the piece of DNA that we're interested in the right spot. That's how it works. Okay, so we've got these two selections here. So it's, as I said, it's not as simple as to say, okay, I do the transformation, I have it all in there. Really, we have to select. A, we have to select for E. coli that have the plasmid, which is the ampicillin, and B, we have to select for the plasmid that has the DNA in the right spot in the plasmid, which is the color. Okay, so you've got these two selections here. So when you get your plate, you'll have colonies that have grown because they have the vector, right? That's the first thing. Otherwise, you wouldn't see them. They would be dead. But then you have to look at the color, of course. So only the white ones are the ones that you're interested in. Okay? Does that make sense? Maybe or not. Most of it. Yeah, anyway, it's a bit complicated, but that's why I'm going through it now, and then we go through it again. Okay, bacterial clones, strain selection. We talked about this as well already a little bit. In your case, you're using a strain called Top 10. It's just a strain of E. coli, they're called Top 10 strain. So as I said, it's been optimized for cloning. And it requires you to know a lot about this. So that's what I said before as well. You can't just use any type of bacteria, obviously. So it has really been optimized and it's commercially available, so we buy that. And when you get your cultures with the E. coli, that's basically what you're getting from us, is that strain. Okay? Okay, and so before we finish, I think that's the last few slides there. Let's talk a little bit about what else we could do when we clone. That leads back to that when I asked you in the beginning, why we're actually cloning, what's this important. So one of the things we can do, of course, is we can make cell lines. Okay, so we can do things like making cell lines. What are cell lines? So cell lines are immortal cells. Okay, they usually have been made by introducing a mutation or infection with a virus, or with a plasmid. Okay, so we can infect cells, we can transform plasmids into cells, and we can study them. Okay, so let's assume we want to study melanoma. Okay, so then you would go and say, oh, how can I study melanoma? Well, I could take tissues from people, right, from patients, and can study that. That's obviously the most realistic situation you want, right? But that's often hard because human cells, they're not very stable, and they need to be cultured, and they can die, so it's very hard to work with that. Sometimes you have to work with them, of course, because if you want to develop some drug, of course, at the end you have to study them in the cells that you then want to look at and you want to treat. But if we go a step back, sometimes what people do is they make cell lines. So you would make skin cells, a skin cell line that you have changed somehow, and that you can culture and grow. I think Graham Jones does that in his research, so he has skin culture cells line that he uses. And so what you can do is with those cells, they basically mimic what happens in skin cells. Okay, apart from the fact that they're immortal, they are kind of cancer cells, but they are kind of mimic the state of what a skin cell looks like. You can then introduce things in those cells. Okay, so you can say what happens if I introduce this protein or this thing, and to introduce that, you will have to introduce it by using a plasmid. Okay, so you can use these plasmids here. You can see they're more complicated, but because they're plasmids, in this case, they're being introduced in mammalian cells. Okay, they're not E. coli cells, they're not prokaryotic cells, they're eukaryotic cells. So they need other things in here. Similar things, antibiotic resistance and other things, but there's also other things in there that you need. Obviously, the origin of replication is different, so on. So all these things will be slightly different when you introduce them in mammalian cells. Okay, so every time you introduce something in mammalian cells, you don't call it transfection. Okay, you call it actually. You can't mix it up. You don't call it transformation. You call it transfection. Okay, so transfection is when you introduce it in mammalian cells. Transformation is when you introduce it in E. coli cells. But the principle is the same. You're basically taking the plasmid, introducing it, making sure that's going the nucleus of your cell, okay, because only it's the nucleus. Remember, the nucleus is the one where you're making RNA. That's where the transcription happens. There's not much point if it goes in your cytoplasm, because there's no there's no RNA being made in your cytoplasm. It's being made in the nucleus. Okay. And so this is one of the examples that we use it for in our lab. We actually use it to make specific proteins. So we have plasmids that we buy commercial plasmids, and we introduce our genes of interest. We clone them in there, so we cut and paste them in there, really, or we do a PCR. And then we're introducing it into those type of vectors. Again, don't worry about the details here, okay? That's really just for information. We're not going to ask you what all these things mean. Just these are more complicated, okay? There are other things in there, and they are being used for other purposes. So in this case, it's a protein expression, okay? So in this case, what is actually in there is, and that's why I mentioned it, because it is interesting here, it's got this fluorescence protein in there. So it's got something in the sequence, okay, before, so you would introduce your protein here, and so what that means is then when you make your protein, so when you introduce that plasmid into your cells, what happens is when you express it, so when this plasmid is being expressed, it makes a protein, but it starts here and it makes a fluorescence protein first, so it makes what we call a tag, a fluorescence tag, so it makes a protein that is fluorescent, in addition to your protein of interest in here, okay? So what you then have is your protein, but the protein is attached to a fluorescence tag, okay, covalently attached. So it's got fluorescence tag and then your protein. So now you might ask, why is this useful? Well, turns out it's very useful, because what you can do is you can then see your protein, right? If you express your protein, you can see, because in this case it's green, okay? And by modifying this tag, okay, so by modifying, this sequence here a little bit, you can go from blue to green to yellow to red, everything. You can make all the colors that you want. And you can make different proteins. You can insert different proteins. You can express them in your cell, and then you can ask the question, okay, if I express the protein, where does it go in the cell? Okay, does it go in the membrane? Does it go in the nucleus? And that's one way of studying that protein, right? If you want to know, is that protein a nuclear protein? Well, then it's in the nucleus. Is it a protein that is in the membrane? And it goes in the membrane, right? And that's the first way you can study these things. So you can see, or you might already know something. You might know this is a protein that is important for the cell walls, in the cell walls, okay? So you then change the protein. You want to know how this protein works. So you're introducing mutation in the protein specifically. You then introducing it in the cell again by transfecting the cell, and you're looking, okay, is it still in the membrane? If yes, well, it's still functional. Then the mutation hasn't done anything, right? But if the mutation changes the protein, and it's not in the cell membrane anymore, then I know this mutation is important. Okay, and that again gives me information of how the protein works in the cell membrane. Okay, so you get the idea roughly? Yeah. So this is the kind of things we use. And we use something else in the lab. We use a protein that is tagged to what's called a GSD. That's another fusion protein. It's not a fluorescence protein, but it's a protein that allows us to then take the protein from the cells. So we use the E. coli to make the protein for us. That then is tagged to the GSD tag. So it's the same principle that this one here, okay, as fluorescence tag, we have a GSD tag fused to the proteins. And because then the E. coli make the protein for us, so we have our protein of interest and then the GSD tag onto it, attached to it. And why do we do that? Well, we do that because we then want to actually purify it. We want to get the protein out of the cell. We want to purify it, so we want to actually extract it from the cell. And then we want to study it in isolation. So we're not looking at the cell. Okay, we're not looking at it in the cell, we look at it in isolation. So we actually make it, it's tagged, it binds, and because it has that GSD tag, it turns out the GSD tag selectively binds to those GSH beads, okay, the little beads that we use in columns, so the columns, beads, and that protein binds selectively to those beads. So that just makes sure that we can purify it, so we can separate it from whatever is left in the cell, okay? So we open up the cells, we lyse the cells, and then we run it through those columns, and the protein binds to those beads, and everything else goes through, and so we have our purified protein. Right? And then we remove the tag somehow, we cleave it or we elute it or whatever, and we end up having the pure protein. So we can then work with the pure protein. Okay? And what we do then is we use the protein to solve its three-dimensional structure. So that's what I do a lot, is we use NMR or crystallography to solve those structures. So we actually look at what these proteins look like. Okay? And this is, you can see here. It's just one example. I put that there to show you one application of cloning, and it's actually the application that we do in our lab. We use that to this is just a whole bunch of flasks here that grow up E. coli. You can see the brown stuff, this dirty looking stuff is E. coli, so you just grow a lot of them. Then you open up the cells, you make your protein, you can see your protein on gels, so you can run that, that's your protein there. Once it's purified and clean, you can then use methods, like I said, other methods they've used to solve those structures. And this is really, really important to understand, of course, right? Because in this case, it's a DNA binding protein, okay? So understanding how this works, just to give you a little bit of insight why this is important is, well, there's, as I said, that goes back to the beginning, there are diseases where you have mutations, like, for example, that one introduced in the PrEC. So you might ask yourself, why is it so that this one mutation is so bad for me, right? Why is this leads to that? And that goes back to the question at the beginning that I asked you, is that one arginine residue could affect, excuse me, could affect protein binding, DNA binding. So if I now have the structure of this, right? Here's the structure. I can see exactly how this protein binds. This is a piece of DNA, in this case, single-stranded DNA, okay? You can see the bases here. You can see each atom in there, so you can see now in here, somewhere in that loop here that binds to the DNA could be that arginine. That sits there and binds to the DNA, okay? So now you can understand, if I change that, of course, then that loop can't bind anymore, and the whole DNA comes off, and it doesn't bind. So then the whole function of the protein, transcription factor, in your case, TP63, in this case, it's another function, which is not important really, but you can then understand why this arginine is important. And that's one thing. The next thing is, what can I do about it, right? Well, I can try to eliminate the mutation in the first place, which is kind of where it goes to in a long-term scenario, where people specifically, and that's, of course, with all the ethical issues that you have, you go in very early and you say, okay, you're taking the embryo and change the sequence and make sure that the mutation is gone, right? So eliminate those diseases from the first place. Or the other thing is, you can develop drugs, obviously, to treat the disease. So in this case, you're treating the symptoms. So you're trying to develop some sort of drug that then helps you blocking certain things. So, for example, if you know that you could help this protein bind somehow by introducing a small molecule, right? But you can only do that when you know the structure. Then you can design drugs to do that. Or the other way, you can design drugs to block certain things, right? But you can also only do that if you understand the mechanisms of it. So if you know how these proteins look like and how they bind, you can design small molecules, drugs, cancer drugs. There's numerous examples, right? Especially for cancer, where people have done this. They develop these drugs, and they work because we know they work because we have solved those structures and we know how these things work, right, in the cell. So that's an example, okay? So cancer drugs is a classical example of those things. And that's where I want to leave it. Are there any questions, guys? A bit earlier today. Any questions, what I just said, or any comments? No. So you can see it's pretty, I'm very excited because that's the research I do, obviously, but it's those type of things that makes me excited anyway in research. Because it helps someone. It eventually will help someone. You design cancer drugs, so you trying to get there anyway, and you will help someone down the track. But this is very basic, obviously, but it needs, regarding this, you need the cloning part to actually get there. Because you remember, you need the cloning part to make your protein, to start your protein, to then design the drugs. So it's a very long process. But you can hopefully understand that why this is important. Any questions? Any questions regarding the lab? The unit itself? I mean, you can always email Lisa. She still responds to emails. Just won't do the lectures anymore. But if you have any questions that you want to ask me, no?
Yeah, question.
Yeah.
I'm not clear on why when we do the use primers and do the first transcriptase to make our cDNA templates and introduce the primers with the mutation. We do two sets of primers and make two fragments and then ligate them back together. I'm not really clear on why we do that.
Why are you doing two sections?
Yeah.
Two sets.
So we're using like two sets of follicle first
Yeah, okay, so maybe I turn off the microphone. It's